So in this chapter, we are going to talk about the history of vaccines, why vaccines are important, what started the whole anti-vax movement, different types of vaccines as far as how they are made. Then we'll move on and we'll talk about the difference between classification and identification. We'll talk about Bergey's manual, and finally we'll talk about different lab techniques that can be used to identify a bacteria. So if you have a patient that comes into the hospital with some type of infection, what are some different tests and techniques that can be used to figure out what the patient is infected with? So that is what this chapter is about. So let's go ahead and continue. Okay, so let's talk a little bit about smallpox. I know we've already talked about smallpox a couple of times this semester. Smallpox is nothing new. Smallpox is caused by a virus. It's been around for hundreds and hundreds of years. They say that as far as viruses are concerned, smallpox has been the deadliest virus of all time. It kills the majority of a population when it infects a population or when it enters a community. At one point, we were vaccinated against smallpox. We are no longer vaccinated for it because it is said to be eradicated from the world. It was eradicated from the United States in the 1950s, and it was eradicated from the world in the 1980s. We have not seen any new cases of it. But as far as a little bit of history, we talked about way back in chapter 1, we talked about Edward Jenner and smallpox. But going back hundreds of years, even before Edward Jenner, the Chinese knew that, or they had discovered along the way, that they could actually take scabs of smallpox patients and they would crush them into a powder and they would blow the powder up their nose. And it made the Chinese immune to smallpox. Now, by today's standards, that sounds very sketchy, very far-fetched. That's not something we would do today. But hundreds and hundreds of years ago, before we had any idea of the immune system or vaccines and how they worked, this was a popular process over in China. And it was very, very effective at preventing people from contracting smallpox if they had had that smallpox scab dust blown up their nose. However, Western society, including all of Europe, was very, very skeptical about this particular technique. So the Europeans did not follow the process that the Chinese did. So the word vaccination is derived from the word vacca, which is the Latin word for cow. Because if you remember moving forward to the end of the 1700s, we had Edward Jenner, who is the person that talked about or had the hypothesis because milkmaids never contracted smallpox. Because remember, there's a closely related cowpox. More recently in the news, there's been the mention of monkeypox. It's all from the same family of viruses. However, cowpox is not fatal for cows, and it doesn't really do a lot to humans. So that is where vaccination comes from. It comes from vacca because of it originally started with cowpox. Okay, so moving forward. So it is estimated that between the years 2000 and 2030, routine vaccines against 10 pathogens, you can see those pathogens here, will prevent 69 million deaths in low and middle income countries. Okay. Now, there are certain diseases such as like measles and rotavirus. Rotavirus causes gastrointestinal illness. It's like diarrhea and nausea, stomach pain. At one point in time, we didn't really hear about cases of the measles in the United States. But we are starting to see outbreaks of diseases such as measles, polio. There was an outbreak of polio recently in New York. So now we're starting to see an outbreak of these diseases that at one point in time we didn't really see. And it is basically due to the people that are choosing not to get vaccinated. Therefore, they're not creating that herd immunity that was talked about in a previous chapter. Remember, you create herd immunity when the entire population is immunized. Therefore, the viruses, the diseases never go away. You're constantly surrounded by viruses and bacteria. But you have been vaccinated for those viruses and for those different bacterial infections, and therefore you don't get sick. Or as in with, you may get sick, but it's very, very mild. So that is the importance of getting vaccine. But there has been a lot of controversy. The controversy, so the controversy is nothing new. You can see the earliest forms of vaccine controversy going back to the 1800s. Okay, we had anti-vax societies forming in England. In the 1900s, in the United States, some of the earliest people that were against vaccines was the idea that the United States was trying to force people to become vaccinated. And that goes against people's right to freedom or free will. People wanted the right of freedom or the right to choose for themselves. And so that again, that kind of started the anti-vax movement. But however, for the most part, people have seen what vaccines have done, how they've saved many, many lives. And for the most part, most people are fully on board for getting at least most of the vaccines. So if we move ahead here to talk a little bit more about these vaccines and what they have done. So in the 20th century, so during the 1900s, the WHO, which is the WHO, the World Health Organization, helped bring vaccines to developing nations, and they have helped in eradicating smallpox, because again, smallpox wasn't fully eradicated from Africa until the 1980s, and this was due to the help of the WHO, or the World Health Organization. Another disease that has been targeted, like I said a minute ago, was polio. Okay, polio was the original vaccine for polio was kind of interesting. People, when they hear the word vaccine, they think needle, that you have to get some kind of needle stick to become vaccinated. That's not always the case. The original polio vaccine was distributed on a cube of sugar. Okay, there's actually a picture of this in your textbook. But they would insert the vaccine, they would load a sugar cube with the vaccine, and you would eat the sugar cube. People would form lines on the weekends at churches or hospitals or empty school classrooms, and they would go and they would get their sugar cube. And that's how a lot of people became vaccinated in the 1950s with the original polio vaccine that was developed by Salk. Polio was considered to be fecal oral, so it was passed in feces. If you didn't wash your hands or there was some type of groundwater contamination, it was possible that a person could spread polio to somebody else. But because most of the people became vaccinated for polio, again due to herd immunity, we saw polio cases decrease pretty much to where they were zero. Until, like I said, recently there was an outbreak of polio in New York, and they blamed it on people choosing not to get the polio vaccines. The problem with polio is a lot of people go completely asymptomatic, meaning they can contract polio and have no signs or symptoms, as you see in this little chart here. 72% of cases, there's no signs or symptoms. However, it is that very small portion where it causes paralysis. And there is a type of paralysis that it causes in some people that causes paralysis of the diaphragm. Well, the diaphragm, this is one of the main muscles that's causes you to be able to breathe properly. It would cause paralysis, or polio would cause paralysis of the diaphragm, literally causing the person to suffocate because they could not breathe. Because of that, there was a contraption invented called the iron lung. Go take a look it up. Last I checked, there was still one person today living in an iron lung. So the iron lung, it was this iron contraption that helped the person breathe properly because their diaphragm stopped working due to the contraction of polio. All right, and you can see in this picture here what it does to polio, or what polio does to a person. It causes deformities. It causes their arms and their legs to bend so it doesn't move right. They look kind of deformed. Okay. All right, so moving ahead from there, we've talked about smallpox. Well, here's a picture of a little boy, it looks like, that has smallpox. You can see the pustules completely surrounding their face and their body, and again, very, very, very deadly. Vaccines have eradicated smallpox. So we don't see cases of smallpox. So again, my personal opinion is that vaccination is very, very important. So what started the anti-vax movement? Going way back to 1998, there was a paper published in this medical journal called The Lancet. The problem, I can look at this and see immediately what the problem was. The problem is that this was a study that only included 12 patients. You cannot perform a scientific medical study with 12 patients. You typically need hundreds, if not thousands, of patients when you're performing a study so that you get accurate results over a large population. 12 people, that's way too small of a sample size of a test population to perform any kind of get any kind of medical analysis or data from 12 people. But it was a doctor that claimed that there was a relationship between the MMR vaccine, which we still get today, and autism. This doctor said, okay, there is a definite link. I've tested these 12 patients that have autism, and there is a link between autism and the MMR vaccine. This is what caused or started the hysteria that we are now seeing. Because soon after this, we started to see the decline of people here in the United States and people over in Europe, namely the United Kingdom, that where we started seeing vaccine rates decrease because people were starting to choose not to get vaccinated because they didn't want to risk contracting or developing autism. It wasn't until quite a few years later, it's the year 2010, that the Lancet retracted their statement. Okay. Now they are saying that based on those 12 people, that was not enough of a sample size, and there was no correlation between MMR vaccine and autism. They've actually went and done numerous studies that had thousands of different patients where they were looking at a link between autism and the MMR vaccine, and they have not been able to find a link between the two. The main author of this paper, he was a medical doctor, but he has since lost his license due to charges of fraud and malpractice based on this scare that he started back in the 1990s. Okay. Okay, so this is what I was talking about, where they've said that they've done numerous studies involving large populations of people, and they found no link. They tested 95,000 children, and they found no link between the MMR vaccine and autism. However, we are still seeing a drop in childhood vaccines because people are still scared of this vaccine, as well as they're choosing, they fear other vaccines could potentially be harmful as well. We're starting to see re-emergences of diseases, measles, chickenpox, polio. Measles is probably the number one disease that I've heard about where there's outbreaks because, again, these diseases haven't went away. They're still in the environment. They're still among the population of people. It's just herd immunity. We've been vaccinated. The entire community has been vaccinated. Therefore, we don't see cases of things like measles, mumps, rubella. But if anti-vax continues on the current route that it's going, we're going to start seeing emergences of diseases that we haven't seen in many, many years, just like now we are seeing with measles and polio. All right, let's talk a little bit about vaccine formulations and how they make vaccines. So here you see that, first of all, vaccines can be ingested. They can be inhaled. For example, there's a flu vaccine that you can get, and you can get it through inhalation or ingestion, like I was talking about with the original polio vaccine on the sugar cube. So when vaccines are made, there's many, many types or many ways that you can actually make a vaccine. There's two main categories that it can be broken down into, and these are the active versus the inactivated particles. Remember, viruses aren't considered to be alive, so you can't really call them living versus dead, okay? However, you can use the terms activated and not activated or inactivated. So activated vaccines, the most common form of the active vaccine is known as the attenuated vaccine. Attenuated is just a fancy word. It means weakened. So some viruses or some vaccines actually contain weakened virus particles. Okay. It means that the virus is present. It's just been attenuated. It's been weakened. Okay. The risk with this is that some people that are immunocompromised, that don't have a fully functioning immune system, may actually contract the disease that they were just vaccinated for. And that is because the vaccine is active. The virus particles have been weakened, but they're not inactivated, they're just weakened. So they could potentially still cause a virus in a person that is immunocompromised. But you can see a lot of the MMR vaccines, there's a lot of vaccines that are currently being made by attenuated vaccines or by the attenuated form but those are considered to be active vaccines. Now, as far as inactivated vaccines, this is where they've taken a virus particle and they've inactivated it. Maybe they've taken off the receptors, or they've removed something that was very, very important for that virus to continue to multiply and replicate. So that virus is no longer functioning. It's a non-functional virus particle. Therefore, it is said to be inactivated. They can also use these inactivated virus particles to create different types of vaccines. Okay, so basically this is what I was talking about with the live attenuated virus. They are weakened, so they are attenuated, so they're weakened. Again, the drawback is that they could potentially cause disease in an immune-compromised host, or even more rare, it's possible, but it's very rare, they could potentially mutate. And if they mutated back to the infectious form, then yes, it would be possible that you're getting this vaccine to prevent a virus, but you wind up contracting the virus instead. But again, that's very, very rare. Okay. And again, with the inactivated viruses, this is where the virus particle has been inactivated by removing something important for that virus. Maybe it's some type of receptor or some type of protein that is necessary for that virus to enter a host cell and replicate. So, but again, the benefit is these are safe for immune-compromised patients because it's inactivated virus. It cannot replicate. It cannot enter into a host cell. So these are safe, and they tend to be stable at room temperature. They're inactivated. So how are they inactivated? Well, you can use either heat. Sometimes you can use chemicals, radiation such as ultraviolet light or gamma rays. Those are different ways that you could potentially inactivate a virus. Okay. Okay, so now we are about to start entering into the second half of this chapter, which deals with identifying bacteria. Two terms, classification and identification. Classification, this is where you simply place organisms in groups of related species, list of characteristics of known organisms. So you're classifying something. You already know its Gram-Stain reaction. You already know what it looks like under the microscope. And so you are simply placing it with other bacteria that are similar. Identification, however, is where you're given a complete unknown. You have no clue what you have. You have to figure out what you have, and then you have to figure out where it belongs. So identification is definitely the more challenging of the two. But identification is what happens a lot, because when you have somebody that comes in sick, you don't know what they have. So a lot of times it's identifying the unknown. And if you look at the picture on the right, that is called a flow chart, or a fancier term for that is called a cladogram. And basically it allows you a step-by-step process. It's basically a process of elimination. So where you start off with a million different species of bacteria, after you go through that flow chart, through the process of elimination, it brings you down to one, maybe two particular species of bacteria. So if you have a patient sample, the first thing you would always do on a patient sample is a gram stain. Always. In every case, you would perform a gram stain. Okay, is this bacteria gram positive or gram negative? And if it was gram negative, then you would follow the arrow down to glucose fermentation. Well, glucose, if you remember, the way glucose fermentation works is the media starts off red and then it changes to yellow, or it can change to yellow with the production of gas. So is acid present, or is acid and gas present? And if you say that acid and gas is present, and you would know that gas is present by the presence of oxygen or gas bubble in the Durham tube, you would go down to motility. Is that bacteria motile? Well, you could potentially stab it into a deep at 37 degrees Celsius and see if it moves away from the initial inoculating line. Well, if it is motile, then you can say, hey, it's Aeromonas hydrophila. Or if it's not motile, then you have to continue the flow chart on the left-hand side. So that you start off this flow chart with quite a few different bacteria species. But through the process of elimination and by performing these biochemical tests, because these are called biochemical tests, like glucose fermentation, if you're looking for if it can hydrolyze urea or if it can utilize citrate, those are considered biochemical tests because it's looking at the biochemistry of that particular organism and whether it can do something like ferment glucose. So you would make your way down the left-hand side of this pathway until you came to the bacteria that you had. Okay, let's talk briefly just a little bit more about Bergey's manual. Okay, so there's two forms. Now, current times, there's two forms of Bergey's manual. There's Bergey's manual of determinative bacteriology, and then there's Bergey's manual of systematic bacteriology. A minute ago, I was talking to you about these 7 books that make up Bergey's manual. That is the first one. That is the Bergey's manual of determinative bacteriology. Before the age of the internet, it was a series of 7 books. Now, because we are constantly discovering new species, while the book is still out there, it's still published, they have made Bergey's manual and they've put it online now so that it can be updated as new species are discovered. Okay, so it stays with fresh new information. But basically, Bergey's manual of determinative bacteriology, again, it's these 7 books. And every bacteria, every archaea that we are currently aware of is listed in one of these 7 books. And then next to the name, it will have the gram stain reaction, the morphology, the cell arrangement, can it ferment glucose, can it hydrolyze urea, can it hydrolyze citrate, everything that you want to know about that one particular bacteria, all the test reactions are there for that particular bacteria. Okay, Bergey's manual of systematic bacteriology, this is the later of the two. This is the newer version of Bergey's. And because this one is based on rRNA sequencing, and remember, that didn't even become a thing until 1978. But after we started getting genetic sequences of these microorganisms, Bergey's manual came out with the book of systematic bacteriology. And this is basically done online. I'd have to look it up. I don't know if this is available in a hard copy for you to actually buy. I know it's available online, and the reason for this is, remember, a typical bacteria cell has about 3,500 genes. The Bergey's manual of systematic bacteriology, it lists the genetic sequence of every bacteria and archaea. So all 3,500 genes are listed. That would probably be an awful big book if it was actually published in a hard format. So that is what makes me think it's probably only available as a PDF or as an online version. But again, it's nothing more than genetic sequences of all the bacteria and all the archaea that we are currently aware of. Okay, so now we are getting into some of the lab tests that can be performed to figure out what a patient is infected with or what they are sick with. So the first one that we are going to talk about, this is actually a really cool one, okay? It's an expensive test, but it's a very interesting test. So first of all, let me start by saying this only tests for human pathogens. Okay, because that's a test question. This test, it goes by the name numerical rapid identification. Sometimes it's known as numerical rapid ID. If you were to look in your lab manual, there is a lab in your lab manual where this procedure is done. It is only good for detecting human pathogens. It's not going to be good for testing pathogens of a cat or a dog or water samples or soil samples or anything like that. Only good for human pathogens. Another thing, you have to have a pure culture. So you're going to have to isolate your bacteria by using a streak plate so that you know you have a pure culture before you can perform this test. The way this test works, the way this clinical lab ID works, is if you look at this tube, I put yellow around it, that tube is called an enterotube. There are 12 compartments. You can look at the tube and see that there are compartments. There's 12 of them. Okay. In each compartment, there is a different type of media. Okay. This one stick, there's 12 compartments, but it can perform 15 different tests because a couple of these compartments are able to perform more than one test. So the actual stick is plastic, okay? And if you look closely at this arrow here that I'm making, right there, that is a metal wire that runs through the entire length of the enterotube. And then on the end, you can kind of see it here in blue on this end, and then on the other end, it's white. Those are screw caps. So what you do is you unscrew the caps on both ends, and it exposes a needle-like point of that metal wire that runs through the enterotube. And you have your pure culture. It works better if it's growing on solid media, such as a Petri plate. And you touch some of the bacteria, some of that pure culture, to the needle. And then if you notice this person's hands up here, how they're gripping the wire, you pull the wire out. And as you pull the wire out, it pulls bacteria through each of the 12 compartments. And then you push that metal wire back through. You put the caps back on both ends, and you incubate the entire tube. What you are doing here is you are performing 15 tests at one time. And so this top enterotube, that's what the enterotube is going to look like before you inoculate it with any kind of bacteria. So after you incubate, or after you inoculate your tube and you incubate it, if you look at the second tube down here, you can see the potential color change that take place. You see, if you're testing for the presence of glucose fermentation, the red media has turned yellow. Okay. If you look down here at urea, it starts off tan. It turns hot pink if it's positive. Very pretty hot pink. Citrate starts off green, but if it's positive, it turns bright vivid blue. So there's a numbering system based on the test, what the enterotube looks like after you take it out of the incubator. There's a numbering system that you use to calculate a 5-digit number. And then there's a website that you go to. You plug in the 5-digit number, and it will tell you what bacteria you have. Okay, so I tested my patient for some bacteria, and my 5-digit code came up to be 21006. Well, I would go to this website, I would plug in 21006 and press search, and it would come up with the bacteria Proteus mirabilis. That's a common bacteria that causes urinary tract infections. So it definitely is a human pathogen. So that is how the EnteroTube works. Now it's great, but it's expensive. I used to order my this particular lab for my students. We would do this in lab. And a box of these sticks, of these EnteroTubes, I think there was like 25 per box. One box is $700. So they're very expensive. However, they're very neat in the ability because you're able to figure out so much information so quickly. Okay, now let's move on and talk about serology. So, serology, we've talked a little bit about serology in the past. Serology is kind of a combination of immunology and microbiology. We're going to be working with antigens versus antibodies, and we're looking for some type of reaction to occur. Okay, so the pathogen will have an antigen. The host, the human, would have the antibody for the antigen. So let's talk about serology. Again, you're combining microorganisms with specific antibodies. All right, so the way this works, first of all, let me start off by saying there are 3 different lab techniques that are all serological based methods. We have slide agglutination, we have ELISA, and we have Western blot. Okay, slide agglutination is seen here. I have a picture of slide agglutination, which I'll go over in a minute. ELISA, remember, that stood for enzyme-linked immunosorbent assay. We talked about ELISA back in the chapter, the virus chapter, because it's used for the identification of viruses. And then there's Western blot. Now, I'm not going to go into the details of Western blot because there's like 12 to 15 steps in Western blot, and it can become kind of confusing. So just make sure for the final exam, you know that these 3 down here, slide agglutination, ELISA, and Western blot, make sure you know that those are examples that use serological based methods for identification. Okay, so let's talk about slide agglutination and the way slide agglutination works. You have a patient that comes in and your patient has been throwing up and your patient has diarrhea and you suspect that your patient has food poisoning. Well, the next question is, well, what kind of food poisoning do they have? Is it E. coli? Is it Salmonella? Is it Listeria? Is it Shigella? So what kind? So what you would start off by doing is you would take a patient sample, okay, and this right here, the background, this is nothing more than a glass microscope slide, a cheap glass microscope slide. You would place your sample on the microscope slide, okay. So in this spot here, originally what that had was just the patient sample. Now remember, your patient sample, if your patient is sick with E. coli, then your patient sample is going to have, you're going to have those antibodies, those antigens for the E. coli in the patient sample. So what you are going to do is you are going to bind E. coli antibodies. So again, you are testing your patient for E. coli food poisoning. If they have E. coli food poisoning, they will have E. coli antigens in the patient sample. So this is the patient sample on the left of the slide. To that, you add the E. coli antibodies. And what happens when there's a reaction? What you're hoping happens is that if you're correct, if your patient does have E. coli, then there's going to be a reaction between the antigen in the patient sample and the antibody that you added to the patient sample. They are going to bind together and they are going to form precipitation. So you see those clumps of white? Those clumps, that is agglutination. Those clumps mean that there was a reaction between antigen and antibody. So whatever antibody you added to that patient sample, then that's the illness or that's the disease that they have. So if you're testing your patient for E. coli and their test comes back looking like the picture on the left, then you can definitely say, yes, my patient has E. coli. However, if you look at the picture on the right, that's a negative test. If you were testing your patient for E. coli and you added the E. coli antibody to the sample and no clumping occurred, that is a negative test. That means your patient is still sick, but they're not sick with E. coli. You don't know what they're sick from, but you know it's not E. coli. So that is the way slide agglutination works. You're looking for a reaction between antigen and antibody. And again, all three of those examples, slide agglutination, ELISA, and Western blot, they all work by trying to combine an antigen with an antibody and looking for some type of precipitation or color change or something which lets you know that there was a reaction between the antigen and the antibody. Okay, moving on to phage typing. Remember what phages are from the virus chapter. Phages, which is short for bacteriophage, these phages are viruses that infect bacteria. So how can we use a virus to determine a bacteria species? Okay, let's recall one thing from the virus chapter. Remember, viruses are species specific. There are viruses that will infect a human, but won't infect any other organism. There are viruses that will infect a cat, but won't infect any other organism. There are viruses that will infect E. coli, but they won't infect any other organism. There are viruses that infect Salmonella or Shigella or Clostridium, and they won't infect any other species of bacteria. So these viruses are extremely species specific. So one way that you can test for the presence of a particular bacteria is with the technique known as phage typing. You add the virus to the Petri plate. You add the virus, and you're looking for an interaction between the virus or the phage and the bacteria on the Petri plate. So just to keep things simple, let's go back and use the example of E. coli. Your patient, you think they have E. coli. So you've taken some E. coli and you've spread it over this entire Petri plate. And you notice that this Petri plate is in grids. You can actually buy Petri plates that already have the grids built in. All right. So you want to test your patient sample for E. coli. Well, there are going to be phages that are specific for E. coli, meaning that virus will only infect E. coli. All right, so after you inject the different viruses, you can potentially inject a different virus into each square, and you can see the virus marks. Those are where the phages have been injected into that particular section. Okay. So you incubate your Petri plate, and then after 24 hours, 48 hours, you get your Petri plate out of the incubator, and you have potentially one of three results. You may have a result that looks like this one. So you inserted an E. coli phage looking for E. coli. Well, it looks like the E. coli phage was successful. It found the E. coli cells. It entered the E. coli cells. Remember what a virus does when it enters into the host cell. It replicates inside the host cell, and in the end, it causes lysis, or busting of the host cell. So that is what that clearing is right there. That clearing is where at one point in time, there were millions of E. coli cells there. but you added the E. coli virus, it infected the E. coli cells and it caused the cells to bust open. So if you get a result that looks like this, then you can say, yes, my patient was definitely infected with E. coli because there was a match between the E. coli virus and the E. coli bacteria. Okay, another potential result that you could have would be something like this. That's a partial match. You could say, okay, yes, my virus, my phage that I entered into that particular area, it was a partial match for the E. coli. Maybe my patient does have E. coli, but the infection right now is very low. Or maybe there was some E. coli present, but they were also sick with something else. So you can see that there is some breakdown of the agar in that particular area, but not the complete breakdown. So that means, yes, there was a partial match between the phage and the bacteria that was added in that particular place. Or you could get a result that looks like this. There's no breakdown. There's no type of lysis that occurred. So maybe you were testing the E. coli phage and you were testing a different patient. Well, there was no breakdown. There was no lysis that occurred. So you injected the E. coli virus into this particular square right here, but no breakdown occurred. What that means is the patient sample that you put in that particular square right here does not have any E. coli in their specimen because there was no breakdown. There was no interaction between the bacteria and the virus. No interaction. Therefore there was no breakdown. And that is how phage typing works. You are using specific phages on specific bacteria to see if a reaction can occur. Okay, so continuing our talk on techniques that are used to identify unknown bacteria. So from this point on, technology has kind of taken over. A lot of the techniques from here on out are basically going to be machine-based. So let's talk about genetics and how we can use genetics to find out the unknowns. Before I talk about DNA fingerprinting, let me go down to the bottom and talk about polymerase chain reaction, or PCR. The reason I want to talk about PCR first is a lot of times you have to perform PCR on your sample before you can find out the fingerprint or the genetic fingerprint of it. So PCR, polymerase chain reaction, this is, again, it is machine-based. What it does is, I want you to think of it like it's a copy machine. It takes one piece of DNA or RNA and it makes potentially millions of copies of that one piece of DNA. Why would you want to perform PCR? Well, a very common reason that you would perform PCR is if you're looking for a very particular species of bacteria, it may be present, but it may be present in very, very low concentrations. So by taking your sample and performing PCR on it, what you are doing is you are amplifying it. What that means is you are copying it. You are increasing the number of copies of that piece of DNA. You need enough DNA to work with, and most tests require a minimum concentration of DNA before you can perform a test on it. So that is what PCR does. It's a copy machine. It makes copies of the DNA of interest so that you can now target it. So there's enough present that you can target it. So again, PCR is machine-based. It takes about 2 hours from start to finish, and it will potentially make millions of copies because it's exponential growth, just like bacteria. So every round, there's about 30 rounds in a PCR program, and every round of the program, the genetic material exponentially doubles. All right, so a lot of time you perform PCR first, and then you take the product of the PCR, and you're able to perform DNA fingerprinting on it. And the theory behind DNA fingerprinting is the same as how no two people are supposed to have the same fingerprint on our fingers. So every species, every organism has a unique DNA fingerprint. So a human DNA fingerprint is going to look completely different than a DNA fingerprint of a cat or a dog or some kind of plant or a tree or a bacteria or some type of archaea. So even between species of cats or species of bacteria, you can look at one strain of E. coli and you can look at a different strain of E. coli, and they are going to have a different genetic fingerprint because they are different strains. So you have 200 different strains of E. coli. Well, each strain of E. coli is going to have a different genetic fingerprint. So you perform fingerprinting on something called, you perform fingerprinting by performing a technique called electrophoresis. Now, electrophoresis can be seen over here on the right. Now, the actual gel, this is actually a see-through, it's a clear, it's a transparent gel that you make. And then if you look really close, these are wells. I'm circling, and they go all the way across. Those are wells that are dug into the gel. And you pipette your DNA into those wells. And then you plug the machine in, and you turn the machine on. And the machine, electrophoresis, works on current. It's negatively charged up here, it's positively charged down here, and it pulls the DNA down towards the positive charge. And as you see, the DNA starts to separate. So if you were to look at this one band of DNA right here, the rule is the smaller the piece of DNA is, the faster it will run on a gel. The bigger the DNA is, the slower it will run on a gel. So this piece of DNA down here is much smaller than this piece of DNA up here. So it takes about 30 minutes for one of these electrophoresis to run. And then after that, you expose it to UV light, and it will show you the band pattern. And so every organism will have a unique band pattern. And so you can take your unknown, perform fingerprinting on it, and then your unknown now has a DNA fingerprint. And now it's simply matching. There is a database of DNA fingerprints where you simply compare yours to the database, and it will automatically find a match for you. Or in the case of a new species, a match won't come back, and that's how you know you've discovered a new species. Okay, moving on to ribotyping. I'm not going to talk too much about ribotyping. Again, it's machine-based. This was the big breakthrough of 1978. Ribotyping is a type of RNA sequencing. It's genetic sequencing of the DNA, of the RNA, and every organism has a unique genetic sequence. Sequencers are not cheap. They're about $100,000, which is why sometimes manual techniques are still used instead of technology, because some of technology is great, but oftentimes technology is really expensive. With ribotyping, you take your sample, and again, it would need to be a pure culture of bacteria. You simply put it in a machine, and the machine does all the work. It would lyse the cells to release the genetic material, and then it would take a small piece of that genetic material and it would code it for you. And because every organism has a unique code, the code that was in your patient sample would match to a code in a database. And as soon as the match was found, well, that would be the bacteria that was in your sample. Again, if no match was found, then you've just discovered a new species. So genetic fingerprinting. So before I started teaching, I was in research. And I spent about 5 years in research, and all of my research was based off of fingerprints, genetic fingerprints, and performing PCR. So it is a very common way that's used today for finding unknowns and for identifying bacteria. Okay. All right. Okay, so let's talk about PCR. PCR stands for polymerase chain reaction. PCR is a technique that I personally hold near and dear to my heart because I did a lot of PCR reactions in the past. PCR, you can think of it as a copy machine. Okay. What does a copy machine do? If you need to make a copy of something, you have a sheet of paper and it has writing on it and you need to make a copy of it and you go to the copy machine and it makes you an exact copy. Well, that's kind of what the PCR machine does. You have a small piece of DNA. The PCR machine makes copies of it. Okay. So you see the different components. I know you had a lab that talked about PCR and talks about the different components that are needed to perform a PCR. This is machine-based. You see the thermal cycler. So this is the PCR machine. In order to run a PCR, you need your DNA samples. Okay. You need your primers, which cut the DNA sample to tell it what section of the DNA needs to be copied. You have your free nucleotides, your cytosines, your adenines, thymines, and your guanines. You have Taq polymerase, which is a very expensive enzyme that allows for the PCR reaction to perform. And then all this is in a buffer, and the buffer with the other ingredients are put into a PCR tube, and the PCR tube is put into the thermal cycler. A typical cycle will run about 2 hours. And within this or I should say within the entire program, the PCR will run about 2 hours. And it typically runs in cycles. Pretty much you can set it between 27 and 32 cycles. So each cycle exponentially amplifies the DNA that's present. So in cycle 1, say you only had one piece of DNA. Well, that one piece of DNA is all great and everything, but one piece of DNA is not enough DNA for you to perform any kind of testing on, because most tests have a minimum concentration that has to be present to detect a virus or a bacteria. So after the first cycle, that one piece of DNA becomes two pieces because it's copied, and it's exponentially copied. So one piece of DNA becomes 2. During the next cycle, 2 pieces of DNA become 4. During the next cycle, 4 pieces of DNA become 8. During the next cycle, 8 pieces of DNA become 16, and so on and so forth. So after about 30 cycles, you have over 1 million pieces of DNA. If you do the math behind it, you will see that. And so that is the importance of PCR. It amplifies a particular piece of DNA of interest, and it makes thousands, if not millions, of copies of that DNA, so that you now have enough DNA to work with. And this is why during the active COVID, they would do a quick test, but a lot of places didn't perform the quick test. They would often perform PCR on it, because what if you had COVID? But what if you only had one COVID virus particle in you? There's not a quick test that would detect that, because one piece of DNA is not enough for that quick test to detect. So they would often perform PCR on it first. The PCR amplified the DNA. So if you only had one piece of COVID virus particle inside you, it would amplify it. So after those 32 cycles over that course of 2 hours, you would now have 1 million pieces of COVID viral particle DNA in that little test tube. Then you would perform gel electrophoresis on it, and you would wind up with a positive. If DNA showed up, or if the COVID virus was present in the sample, it would glow as a band. So you would always typically perform PCR on your sample to copy it, and then you follow up with gel electrophoresis to see if that band of DNA or RNA, because it can detect both DNA or RNA, to see if that band of genetic material is present. So that is what PCR does. It's a really great technique for amplifying DNA. Let's move on and let's talk about nucleic acid hybridization. All right, nucleic acid hybridization. So again, nucleic acid means that we are dealing with DNA or we're potentially working with RNA. And it compares DNA from two organisms. You have to have two organisms present. And it compares the DNA between the two organisms to see how well they match up. All right, so the way this test works, or the way nucleic acid hybridization works, is if you look over here, you have organism A DNA. So you have that, whatever, that bacteria that could, let's use staph. Let's use Staphylococcus aureus. Okay, so organism A DNA, I know what it is. It's going to be Staph aureus. Organism B DNA, I don't know what that is. That is my unknown. I'm trying to figure out what organism B is. So one way I can do that, the purpose of nucleic acid hybridization, is to compare the genetic sequence of organism A and organism B and see how well they come together. All right, so the first step that has to happen, remember, DNA is double-stranded. Those double strands are held together by hydrogen bonds. The easiest way to break a hydrogen bond is with heat. Okay. So you take a sample of organism A, which we know is our Staph aureus, and we take a sample of our unknown, we add them to the same tube. We put this tube in a particular machine, and the first step that the machine does is the machine heats up the samples. It heats up enough to break the hydrogen bonds that are holding the double strands together. So at this point, you have single strands of your Staph aureus, or your organism A, and you have single strands of your unknown. Well, remember, they're all in the same tube together, and they're floating around, they're mixing together. So potentially what's going to happen is some of your organism A DNA is going to interact, it's going to find its way over to organism B, and it's going to mingle with organism B. And so you have all these single strands floating around this tube, and then at some point what the machine will do is it's going to start cooling back down again. When the machine cools back down, those hydrogen bonds reform. So those double strands attempt to come back together. And you will wind up with one of three results. You may come up with the one on the far left, which is complete hybridization. And if you look, it's got one strand of the dark purple DNA and one strand of the light purple DNA. And it's a complete match. What that means is there's complete hybridization between organism A and organism B. Well, we know that organism A is Staph aureus. So because there was complete hybridization, that means that organism B also has to be Staph aureus. Okay, another potential result that we could get is the one in the middle, which is partial hybridization. That means that the organisms are related. They kind of matched up. Okay, so you took one strand of organism A, one strand of organism B, and you get this middle result, partial hybridization. So what that means is, well, we know organism A is Staph aureus. Organism B, what if it's something similar? What if it's Staph epidermis? It's similar, it's the same genus. It's just a different species. So they are related. So you would have partial hybridization there. Okay, or you could have the one on the far right, this last potential result, meaning that there's no hybridization that occurs. Organism A is completely different than organism B. We know what organism A is. We still have no clue what organism B is. We know what it's not. We know it's not any type of staph, but we don't know exactly what it is because no hybridization occurred. Okay, for the exam, I want you to make sure you know that there are 3 different tests that use nucleic acid hybridization: Southern blot, DNA chips, and FISH. Okay. A minute ago I talked about Western blot, and I said we weren't really going to talk about Western blot because there's like 12 steps and it's very involved. It goes into a lot of detail. Well, Southern blot is the same way. It's very involved. It goes into a lot of detail. So all I want you to know about Southern blot is that it is a type of it uses nucleic acid hybridization, but you don't have to know any of the details. Okay, so I have 2 slides left. Let's talk about DNA chips, and then let's talk about FISH. Okay, so DNA chips. This is considered to be the latest and greatest in technology. With DNA chips, you have the ability to test an unknown against potentially hundreds of thousands of genetic sequences at one time until you find a match. The way this works, if you look at step A, you have a glass slide, you have a barcode at one end because, again, this is machine read. The other side, you have some dots, and it looks like they're just dots. However, if you read what those dots are, it says that those dots contain hundreds of thousands of synthetic single-stranded DNA sequences. That's pretty impressive because all it looks like to us is some dots. Okay, B, you take the patient sample. You take this sample, but remember, patient sample, our DNA is double-stranded. So once again, you have to separate it into single strands. And DNA can be very large. You can chop it up to make smaller pieces out of it, which is what they're doing in step B. They are separating the double strands into single strands, and they are chopping it up to make it into smaller pieces. And then they add the DNA from step B to those chips on the slide, or to those dots on the slide in step A. And that's kind of what you see here in step C. You see some partial hybridization occurring, or a little bit of hybridization occurring between the unknown and the known, which is on those chips. And then you insert it into a machine, and the machine reads it. You have several different potential outcomes. If you have a green dot means that there was complete hybridization between your unknown and the dot or the single strand that was on the chip. You could have yellow or orange. What that meant is that there's partial hybridization between the unknown sample and the DNA embedded on the chip. Lastly, you could have red. Red means that there was no hybridization between the unknown patient sample and the DNA on the chip. So by doing this, you can compare one unknown against hundreds of thousands of different bacteria species at one time. So it's supposed to make the process of isolation and identification much quicker than traditional methods. Okay. Moving on to my very last micro slide for you guys. This is a process known as FISH. FISH stands for fluorescence in situ hybridization. FISH has been out since the 1980s. We've been using FISH for a very long time. Originally, FISH was not used in microbiology. FISH was originally used to study cancer. The thing that was so great about FISH is in the name, fluorescence in situ. All right, let's talk about in situ and what it means. There is in situ and there is ex situ. Normally in the early 80s or in the late 70s, before FISH was developed, if a patient had cancer, they would do a biopsy of the cancer, and they would study the biopsy in a lab under microscopes. That is considered ex situ, outside the body. When FISH was developed for the first time ever, we could leave the cancer inside the patient to study it. That is what is known as in situ, or in the patient. Cancer behaves differently. Once you cut it out and take it to a lab and study it under a microscope, it tends to behave differently than how it would behave in its host. We wanted to study it in its natural environment so that we could understand it better. And that is what is so great about FISH. It allowed us to study the cancer while it was still inside the host. That is what in situ means. All right, so with FISH, It combines fluorescence and it combines probes. A lot of times the fluorescence molecule, it can be red, it can be yellow, it can be green, it can be purple, it can be any color you want it to be. And you attach it to a probe. And this probe is designed to attach to whatever it is you're studying. So if you're studying cancer cells, the probe will enter into the body through either injection or sometimes through swallowing a liquid. The probe will circulate throughout the body and it will bind accordingly to what it's designed to bind for. In the case of cancer, it was designed to bind to cancer cells. In the case of microbiology, you might be looking for a particular disease or particular bacteria. Is there any salmonella in a patient? Well, you can design a probe that's going to bind to salmonella. And then you can inject the probe, and the probe will circulate in the body, and it will bind to any salmonella. And then you give them a CT scan, and any of that salmonella is going to glow. So you can look at that patient scan and you can identify what bacteria is present by whatever you decided to test for. And that is what is so great about fluorescence in situ hybridization, or FISH. We still use FISH quite a bit today because a lot of times we want to study organisms or we want to study pathogens in their natural environment. So that is why FISH is oftentimes still used today. All right, and that is all I have to say about FISH. And that's all I have to say about this entire chapter. And that is microbiology for the semester. I am completely done with lectures. Your final exam again will cover chapter 16, chapter 20, and the first 9 slides of chapter 15. All right.